Hi, my name is William Bauer. I'm a high school science teacher, OpenSciEd facilitator, and curriculum writer. Let's dive into Lesson 8, one of my favorites of the Cancer unit. How did we get here? Lesson 8 falls about halfway through this unit. During the first half of the unit, students figure out why older and taller people get cancer. As we age, we accumulate mutations due to random errors that occur during DNA replication. If those mutations affect the function of the p53 protein, then we may develop cancer. After learning about p53, students may be left wondering why cancer seems to run in families, or how a young person who hasn't had their DNA replicate as much could be at risk of developing cancer. This leads us to investigate this question in bold. How can cancer risk be inherited? Let's look at this lesson at a glance. This lesson is 2 days. There's minimal preparation involved in this lesson. You'll need beads and pipe cleaners to model inheritance and meiosis. There's a reading to print off and a few handouts. And it's important that you preview the videos of Lauren and Lakita, two young women who've developed cancer at a young age. In part 1, you'll analyze pedigrees and model them with pipe cleaners representing chromosomes and beads representing alleles for genes that affect cancer risk. On day 2, you'll read about meiosis and revise your pipe cleaner model to take into account how crossing over can result in new allele combinations. More about the first part of this lesson. On day 1, we're introduced to two young women who I happen to interview who have an inherited gene mutation to their p53 gene that leads them to develop cancer at a young age. They'll share their stories. After you learn more about their personal cancer history, you'll analyze the pedigree of one of their families to investigate patterns of how this gene mutation has been passed down through generations. We model these patterns of inheritance using pipe cleaners to represent chromosomes and beads to represent different alleles related to cancer risk. On day 2, you're introduced to a more complex pedigree that involves two traits, two different genes that are involved with cancer risk. Students identify that there are some new allele combinations that weren't present on the parental chromosomes, which leaves them a little puzzled. In the second part of this unit, or this lesson, we use a reading to learn more about meiosis and to revise our pipe cleaner model of a pedigree to take into account what we learned from the reading and how crossing over works. And that these chromosomes have the ability to swap sections during meiosis, resulting in new allele combinations. So in all, we figure out that pedigrees show that occurrence of traits in families and can help model inheritance, that during meiosis, chromosomes can swap regions of DNA in a process called crossing over. And this phenomena allows for new allele combinations to form and increasing overall genetic diversity. And we'll think about this in the context of cancer risk. Here are some key moments. In part 1, you'll learn more about, you'll be introduced to Lauren and Lakita, two young women who have very interesting stories of developing cancer at a very young age due to having a genetic inherited disorder to the p53 gene called Lee-Fraumeni syndrome. We see how this trait gets inherited through Lakita's family in the pedigree shown in the slide below, and use pipe cleaners and beads to show how what chromosome and allele combinations members of Lakita's family must have. At the end of day 1, we are introduced to a more complex pedigree that has two genes on it, BRCA1, which affects breast cancer risk, and p53, which students are very familiar with at this point in the unit. Highlighted here in red, you'll see that person 4 and 5 have chromosomes that have allele combinations not found in the generation above them. This motivates students to revise their model after reading about how sections of chromosomes can swap places during meiosis, resulting in new allele combinations, such as seen in person 4 and 5. At the end of this lesson, students take an exit ticket to assess their knowledge of meiosis and crossing over. This exit ticket also leads students to revisit their questions about how environmental factors lead to mutations and increased cancer risk, making a good segue to our next lesson. Here are a few tips about managing materials during this lesson. It's really important that you preview the videos of Lauren and Lakita ahead of time. In addition to their family histories, students will watch additional interviews detailing their cancer treatment during lesson 11 of this unit and the support systems that they had while going through cancer treatment in lesson 12. Students hearing these experiences of young people close to their age having cancer may spark new and strong feelings in them. This would be a good opportunity to revisit your community agreements that you formed at the start of this unit before you play these videos, because what Lauren and Lakita share may bring up strong emotions in your students. When you get into looking at the pedigree, it's important to distinguish that Lauren and Lakita have inherited mutations in their germline cells, not from, their P53 hasn't been mutated from accumulations they've accumulated during their lifetime. Another important tip is to color code your beads with the slides that you present so that everyone can be on the same page. That'll make your job easier going from group to group to compare student thinking. And if you have students visit other groups to look at their pedigree models, they can all be on the same page by having the same colors representing the same alleles for the same genes. In this lesson, students will encounter the word inheritance, which they may be familiar with from middle school. The reading will go over the definition of crossing over, and students will have the opportunity to model the crossing over process with their pipe cleaners and beads. Leaf-Romani syndrome, osteosarcoma, MDS are words that we encounter when we learn about Lauren and Lakita's cancer journey. Meiosis and allele are two words we co-construct in this lesson. There's no new posters created in this lesson, but it's important at the start of the lesson to revisit your driving question board, because you're kind of transitioning into this new part about inherited mutations instead of having learned about mutations that accumulate during one's lifetime. And then at the end of the lesson, it's important to revisit your driving question board, because students may have questions from lesson 1 about environmental risk factors related to cancer. Those will be addressed in the next lesson. The next lesson is about how genes interact with the environment to affect skin cancer rates. So thank you for listening. I hope you enjoy teaching lesson 8 with your students. Thank you.